Yep.
Okay, I've got the session recording now. Okay, go ahead, Mandeep.
Uh,
Do you have a question?
yeah, Dr. Terry. So my question is like pretty general. So like for this exam one, like what
Uh-huh.
should we focus on the course wise? So like yeah, we are I want to know what topics what things we should cover for this thing, because I am doing very bad at the quiz, so I'm extremely terrified
Okay.
for this exam. So yeah.
Okay. So now I use this time for all three of my classes, and there's a little bit of overlap between some of them, especially the 4330 and the 6330. But which course are you talking about? The bioevolution course, the 3301?
No, like it's 6330.
Sorry. Okay. Yeah. So you're the grad course. So um, for my undergrad courses, for each of the lectures, I provide what I call an SLO, which is a student learning outcome. And it's basically highlights, notes, little reminders about things that you should have learned or gotten from the lectures. My suggestion, and I don't provide those for the grad courses, just because I think part of the process of a grad student is you learning kind of how to do that yourself, what material is critical, what's important. And so as you're going through, hopefully you've already taken some notes, but as you go through the different lectures, you should be basically making short notes, outlines for yourself, and then you can fill them in with a little bit more detail. And that way, if you've got your own kind of outline, major things that are talked about in the lecture, you don't need to spend a ton of time filling in. And if there are any gaps, like, oh, I've got this down, but I don't quite understand it perfectly, then you can come to me and fill in and ask questions. So that's what I would recommend from the grad course. And if you're having trouble on the quizzes, this applies to all of them. If you're having trouble on the quizzes, you can go back and review your answers. I just posted, I think a lot of people didn't realize that they could do that, but you can go back and review your answers on quizzes and make sure you understand kind of what the correct answer was. And if you're not sure why that's a better answer than the one you chose. And sometimes, it's obvious. You're just like, I just had no idea. Other times it's like, oh, I didn't quite understand it. So make sure you understand why you missed the question. And if you don't, then, bring that to me and say, what's going on here? Why is this a better answer? And so as you're preparing for one of my exams, if you've got the SLOs for the undergrad courses, then use those, rely on those heavily. Those give you a really good outline of what's going to be on the exam. And as a grad student, you can kind of build those yourself, and then you have them to go back and review. And then the last thing I'll say, again, this is maybe a little bit late now for unit one, but you could certainly use it for the coming rest of the semester, is um, it's really, I think, valuable for most students to handwrite notes. Um, it's easy, especially for online courses, to either type notes or just sit there and listen. But you'll find, this is, again, not everyone, but 90% of students, I think, will learn and remember things much better if they write out the notes. And then if you look up videos online, there are even little strategies about how you organize your notes, or, drawing a quick picture will help that muscle memory to remember what's going on. So those handwritten notes, I think, can be very valuable. So those are my general suggestions for getting ready for the upcoming exam.
Okay.
Okay, Vanessa, do you have a question you want to jump in?
So I have yeah. So I have my sorry.
Yeah, I went ahead and I was looking at how I did get the answer correct for the maximum likelihood, but I was kind of interested to know a little bit more as to why is I think it was the heterogeneity, why is that like kind of a weakness? Because when I looked at the graph, it looked like the more you let it run, like the more accurate it could be. And so I just wanted
Yeah.
to get like maybe I misunderstood it.
So it's becoming less of a problem now, but originally, when you picked your model of evolution, you had to pick, let's say, a substitution rate. How often does an A turn into a T? Right? And so you would estimate how often that occurred by looking at a preliminary phylogeny, and then using a quick algorithm to kind of estimate, okay, we have this many A mutations turning into, adenines mutating and turning into thymines. And then you would use that value as part of your analysis. But if there are different parts of the same gene or different genes where that value might be different, then suddenly you're using an inaccurate value, or you're getting kind of a middle ground where it might be higher for one part of the gene and lower for another one. And so that's what we mean by rate, the heterogeneity or the heterochrony, or what is it? I forgot. Is it it's not heterochrony, it's tell me the term again, Vanessa. It slipped my mind.
Yeah, let me pull it up right now. I just had it open because I was looking at it because I was trying to figure out because that was a really tough question. Like I picked the statistics one because I was like, well, maybe not everyone's good at math. But then again, like I felt like it was really circumstantial because there are people that are really good at statistics, and of course, maybe you do have like a lot of good data ahead of time.
Yeah.
Well, let me see here.
I'll see if I can find it also. I think I've got it
Yeah,
here
I'll,
on the SLO
here,
for the 4330.
here we go. I have it. It says here they are inconsistent over some ranges of evolutionary rate, heterogeneity, also in parentheses, heterotachy.
There it is. That's the term, heterotachy.
Heterotachy.
And heterotachy is just, so tachy refers to rates or so how quickly the DNA is evolving. And so originally, if parts of your data set were evolving and accumulating mutations faster than another part, that was a big problem for the maximum likelihood, because you would either pick.
But for.
Sorry, go ahead.
But for like future, but now currently that's considered not a big weakness anymore because obviously like we're better at organizing and looking at our data ahead of time now, right?
Right, and it's not so much the organizing as it is we have more complex algorithms that allow for different models of evolution across different partitions. Different parts of the data can have a different model of evolution, or the same model of evolution but with different parameter values. The trade-off to that, though, is now you've got that much more uncertainty because you're estimating more parameters, and it's also more computationally intense. But yeah, so that's less of an issue for modern analyses.
Yeah.
Dr. Terry, how much do we need to know about neighbor joining and maximum likelihood and Bayesian for BioEvo?
So we don't get into all of the details about it for BioEvo. So BioEvo, you need to know the parsimony, right? And how to map characters onto a phylogeny, and then be able to choose between, if you have 2 or 3 simple phylogenies, be able to choose which is best based on that mapping of characters. And then for the, and my 4330 students and grad students are going to be jealous of you, but for the maximum likelihood in Bayesian, all you need to know is those are alternative ways using statistics to analyze a phylogeny. And that's it for the 3301 course. Okay, Uriel, you can jump in there.
Yes, I had a question. I'm from the graduate course, and
Okay.
I had a question just on the last. I guess I'm asking for your advice on this one. On the last quiz regarding, this was the only one question that I had like a lot of trouble with, which is question number, the last one, regarding the most likely phylogeny. Like I understood that the best phylogeny exhibit homoplasy in one character, but how do you distinguish with this synapomorphy, like for the second answer?
So a synapomorphy is simply,
like how do you see it?
So a synapomorphy, the definition of a synapomorphy is a character that evolved once in an ancestor, and then none of the descendants lost it. So it's still a defining feature of all of the descendants for that ancestor. Right. So we introduce synapomorphies as kind of morphological characters. So for instance, mammary glands are great synapomorphy for mammals because all species of mammals still have them. They were evolved in the common ancestor. Now, from the genetic perspective, those can be just a single DNA mutation that maps to the ancestor of one branch, and all of the species in that branch still have that. So that's a synapomorphy. So once you've mapped your characters onto a phylogeny, any character that you've marked on only one place, that by definition is a synapomorphy for the group. You know, the group whose ancestor you've marked that character onto.
Okay, so for example, I know that the reason why I had so much trouble with that one specifically is because I don't know if you remember the phylogeny or how it looks. I know DNF branch from the same common ancestor, and on the quiz they both had an adenine, and all of the other and they were the last on the I guess the last clade or the last part of the tree, and every other character had a tymine. So that means that it's as an apomorphy because DNF share a common ancestor, and that's the
careful. It's a potential synapomorphy, but the only way we know it's a synapomorphy is when we map it onto the phylogeny and then decide. And that was, I think, part of that question, right? So you're looking at question, and this is from just to be sure, because I pulled
the last one, 5, I believe it's 5.
Well, they're in different orders on different quizzes. So but yeah, so it's the one that says refer to the matrix and the best phylogeny from the previous question and mark all of the statements.
Yes.
True. Okay. So on the very best phylogeny, so first you have to figure out which of the phylogenies is best, right? So for instance, which character were you looking at? The very first character is the one you're talking about, that D and F share a characteristic. So that's a synapomorphy on a phylogeny where D and F are most closely related to one another. However, if you're looking at one of the other phylogenies, then it's not a synapomorphy. If D and F are distantly related to one another, then that has to be explained by either convergence, right, evolving separately in different lines, or as a simplesiomorphy, where it happened in an ancestor but then was lost in one or more of the descendants. And so you can't just look at a
perfect. That was that.
You got it?
Yes, like that was my main. I had so much trouble because I thought in my head I went with the synapomorphy. Well, everybody in the matrix will have it. So that's what was my error, thinking that all of them A, B, C, D, and F should have it, and it was only D and F.
That's the question.
The common ancestor.
Yeah, and so that's why you'll always, if you're ever being asked about a synapomorphy, you'll be given, you have to have three things. You have to have the characters, of course. You've got to have a phylogeny to look at. And then you have to have which group on that phylogeny are we trying to determine, because that would be a synapomorphy for E and F, but it wouldn't even be a character for any of the others on that best phylogeny.
Okay, perfect. That explains it all. Thank you very much,
Awesome.
Dr. Terry.
Yep, no problem.
And one quick question. Should we focus on the readings for the exam, or is it like half reading?
Yeah, for the grad class, the readings are a little more important, but don't feel like you need to memorize them because they get technical and they're in depth. It's a lot, and I know that. So my approach would be to read them, skim over them at least, but don't read them in a ton of detail. And then use the lectures as a guide to what level of detail you need to know.
Perfect. Well, I appreciate your help, Dr. Terry.
Okay. Yep. No problem.
Doctor, I have a follow-up question with what he said regarding, mapping it. How do you determine phylogeny?
Homoplazy?
Yeah.
Let me just, I'll share the example he was talking about so you can see it. You find the link here. Here we go. Okay. Okay, so this is the question he was talking about. It's different than questions you've seen. Maybe it's a little bit more detailed because I'm asking a whole bunch of questions and they had to evaluate every single one. But basically, I gave them a matrix, and then they had to map the character onto these three phylogenies and figure out which one was the best one. So that was the first part. Okay, so do you feel comfortable doing that, or do you want to practice it?
For the best one, it's just like the simplest explanation is like the best one.
Right. So if you know how to map all the characters, and these are fairly easy, simple characters to map. If you know how to map all the characters, then all you do is do that on the three different trees, find the simplest one with the least number of characters mapped, and that's your best phylogeny. Okay? So it turns out that once you do that, if you go through that process, this phylogeny C is the best explanation. It's the most parsimonious. So that's our best phylogeny. Okay? So that's what we established question 2. And then he was asking about question 3. So how do we figure out what characters are synapomorphies? Well, we look at where they're mapped. So the first character in this matrix says that D and F, they've got an adenine. They're different from all the others which have a thymine. Now the best explanation for that, let's just do a quick map. And Word's not ideal for this, but it works. Boom. So we would map a little change on that branch right there, because that's the simplest explanation for why D and F share that same character if they're closely related to one another. Okay? So that now, by definition, is a synapomorphy for D and F. But if we're going to map the character onto this phylogeny, notice D and F are not closely related to one another as they are over here. So what we have to do is map it on that other phylogeny twice. So we would put it, one of them would go on, this would be convergence, and this is the simplest explanation in this example. We'd have to put one of them on the branch leading to D and one of them on the branch leading to F. And so on this phylogeny, that's a convergent character, which is one of the forms of homoplasy. It's a less simple explanation than a synapomorphy. And actually, this is one of the reasons why this tree is not as good as this tree. We look at all the characters, of course, but right now we're saying, oh, I like this tree better overall because I don't have to infer this less likely event that it was a convergent mutation rather than a ancestral mutation that everyone still shares. And so to determine whether a character exhibits homoplasy or whether it's a synapomorphy, you need to have the characters, of course, you need to have them mapped onto the phylogeny, know which phylogeny you're talking about. And then you just look and say, did they happen once and everyone still has them? Did they happen once and a few of them lost it, which would be symplesiomorphy, a form of homoplasy? Or did they happen twice independently, like over here, which is a form of convergence? And there are sometimes examples where we can map a homoplacious character as either a synapomorphy, either a symplesiomorphy or a convergence, but we can't distinguish which one. But on this tree, it doesn't make sense. It's less parsimonious to map it as a symplesiomorphy. You'd say, oh, maybe the mutation happened here, but then it would have had to switch back in E and also switch back in C. And so that's 3 marks. So we don't like that. So it's much better to map this character on this phylogeny as a convergence rather than a symplesiomorphy. And this is, I know this is kind of advanced, especially for the 3301, but this is about as complex and advanced as we're going to get. So you should be able to map characters onto a phylogeny. Once you do that, determine which phylogeny is best, and then be able to determine whether the characters are synapomorphies or convergence or symplesiomorphies.
Got it. Thank you.
Okay,
So
Vanessa.
for question 3, I'm so sorry.
That's all right. Go ahead.
Yes. For question 3, okay, so B, that's actually a pretty good argument saying, like, you know, looking at all, like, every position kind of like talks to you about the evidence. So why wouldn't that be a good idea to look at?
So it would be, and in if it was not a parsimony analysis, this actually would be a valid assumption in one way, because for a model of evolution, we use every single position to help us pick our parameters because it tells us how often mutations are occurring. But for a parsimony analysis for a parsimony analysis like this one, we don't. All we do is look at it and say, oh, character 2, for instance, everybody has a C. That doesn't give me any information about relationships. And then number 2, number 3, there's no information for parsimony analysis for characters 2, 3, and 4 because all the species are the same. So it doesn't give us any information about relationships.
Okay.
And there are actually 2 types of characters that are non-informative in parsimony. One is one that's completely conserved like this. And another one, which we don't really do in the 3301 class, but we call them an apomorphy. So it's like a specialized type of a synapomorphy, but an apomorphy is a character where only one species is different from everybody else. And that doesn't give us information about relationships, because all it does is confirm what we already know, is that, oh, that's a different species. It's different from everyone else, but it doesn't help us to figure out what that species is related to.
So I'm sorry, just to reiterate what you said. So an evidence of this actually being useful to do would be in a, what'd you say, an anasomorphy? Like that's where we should actually take into consideration every position?
Oh, so what I said originally was, if we were doing a maximum likelihood analysis, we would actually use, we would consider every single position. Now, we wouldn't use this as data because that doesn't really help us a lot, but we can use it to help us figure out how often mutations are occurring. Because if we're saying, oh, look, here's evidence that a C mutated into an A in character number 5, and then we see that there's a certain number of sites that are C's already, that gives us an idea about how common this mutation from a C mutating into an A is. So all of the sites help us to inform our models of evolution for the maximum likelihood and Bayesian analyses, which for the undergrad courses we won't do until the next unit. And in 3301, we really don't even do Bayesian analyses. We just mention that they're a statistical form of phylogenetic inference. But yeah, if this was a question based on a maximum likelihood or Bayesian analysis, then yeah, every position would provide, even though they may not provide direct evidence for relationships, they would provide useful information for helping us decide rates of evolution and models that we should use.
All right, thank you.
Okay. Christina or Niharika, if you want to jump in, please unmute yourself and ask a question, or you can just kind of follow along. What else, guys?
Dr. Christina asked something in the group chat.
Oh, sorry, I don't have my chat open. Thanks. Okay, so Christina asks, would it be possible to get partial credit for question 3? So for these other ones, most of my multiple choice or multiple answer questions, I don't give partial credit. It's just kind of an extra level of difficulty. Potentially, you can actually, it will be graded with partial credit. So if you only selected this one and you selected A and then didn't select any incorrect answers, then you could get partial credit. But if you select one correct and one incorrect question, for instance, then you don't get partial credit on it. So yeah, they're already graded the way they are. And I do recognize those are a little bit higher level of difficulty, and there aren't very many of them usually on exams, but there will be some.
Hi, so I actually had a question about the last quiz, and I'm in the molecular evolution class.
The 4330?
Yes.
Okay. Let me pull it up and I'll put it on the screen.
Wait, Dr. I gotta go, but it was nice talking. Thank you for the answers.
Yeah, let me know, Alan. You can take the test. Yours is today, right? Your class?
Yeah. Okay. Yeah, I need to study for like a little bit more. Shouldn't be that bad, right?
No, I hope not.
Yeah.
So yeah, you won't know until you take it though, right? But yeah, if you do have any questions, email me. I'll be on email pretty much all afternoon.
I was gonna tell you. So I took the MCAT recently, and one of the passages was about Bitcoin, and like the passage was super easy for me because I knew the material, you know.
Oh, good. Yeah, I had another student. I don't know if that's a trend, but I had another student who took it about a year ago and said that they had like an entire section on evolution and that they felt pretty confident about it. So good. They do test kind of widely for the MCAT. It's not just medical based or chemistry stuff.
Yeah.
Okay.
Yeah.
All right. So here is Neha. Was it you who asked the question on this quiz?
Yes.
Okay. So was there a specific one
Okay,
that you
so
were?
I was, yeah, so the one about inferring the true tree,
Okay.
I was a little confused, I think, probably on the wording of that, because I think in the videos, I remember hearing something about like, if we do find like congruence between the parsimony and the maximum likelihood, then
Uh,
we could like, that would be a little bit more affirmation, I guess, like that it would be the
yes.
tree, but
Yeah, you'd have a little bit better chance of it. So I guess maybe the best analogy for this question is if you really want to find out what truly happened at a event that you weren't on, right? Let's say you're a policeman gathering evidence or data about, you know, what happened at a crime scene. And if I said all you need to do to find out what really happened is ask two people, and if they both say the same thing, then that's 100%. That's the true thing. That's what happened. So although, yeah, you can be more confident that that's what happened or that's the set of relationships if you have congruence between methods, you never know. And that's the key here is you never know the true tree for any species relationships unless you've actually watched those speciation events occur. Right. And so that really doesn't occur outside of maybe some very carefully curated, even like viruses or bacteria, which, you know, we could in our lifetime have significant evolution in trees that have evolved from those. But unless we're doing it in a lab under very, very careful circumstances, we still don't know exactly what the true tree is. We're hoping that we're finding it, but we never know for sure. So that's the key there, is that inferring the true tree for any set of species relationships is really not an option, although congruence would give us a little bit of a better hope that we had it, or at least we're close to it.
oh, okay, so that key, I guess, was the part where it says all you need to do is that.
Yeah, or maybe even more so, this is kind of reinforcing or testing you on this idea of finding the true tree for any species, right? Which is the real answer is we'd never know for 100%. So there's really no such thing as quote unquote a true tree, because we cannot invent a time machine and go back and know for sure that that's the true tree.
Okay. And I also was um, a little confused with the one about the phylogenies, where every character can be mapped as a synapomorphy.
Okay. So if we have a phylogeny
So I,
where everything is mapped as a synapomorphy, that means we can put every character in just one place. We don't have to infer any sort of convergence or simplesiomorphy. And so it's kind of, again, making sure you know what that definition means. So by definition, it would be the most parsimonious tree because that's the shortest tree you could possibly imagine, right? So we can eliminate A, because that is a correct statement. C, you would need to resort to Fitch optimization to map characters. So Fitch optimization, and we don't do this in the 3301 class, but for the grad class and the 4330 class we do. That's a way to help you find the most parsimonious mapping for very, very complicated characters. But if it's a simple synapomorphy, you just find the branch that includes all of the species that have that characteristic, and you map it. So you don't need Fitch optimization, right? So
that was why
oh, did I
I
make?
was confused between that answer choice, because it said which is not a correct
Oh, you're
statement.
right. You're right. I should give credit for C also. I miss. I should have changed that and said you can map. Let's see. You would need to resort to Fitch optimization to map characters. You're right. That's not a correct statement. I'll go back through. Thank you. You caught that. I'll go back through and I'll give credit to everyone who put C also, because that should have also been a correct answer.
Okay, thank you. Yeah, because I was confused. I was
And in
thinking that
fact,
you
it
did.
even looks, I must have just, I probably should have said, anyway, I needed to rephrase that. It looks like D also, because synapomorphies can be either It could include, oh no, you're right. So D is a correct statement, but yeah, I probably could have simplified the way that that question was presented. So I'll go back in and give credit for C also.
Okay, thank you.
All right. All right, Christina, Vanessa, Mandeep, you're still here. If you have any questions, just unmute yourself, jump in.
Can we like engage a little bit more in this lymphomorphy and like look into quiz 2? Can you open that up?
For this is the molecular evolution course?
Yeah, for the graduate course. Yeah.
Okay, let me get it here. Quiz 2. Okay.
All right. So for the number 1, for there to be an actual like symptomorphy character 1, because it's at the bottom, it wouldn't work. Can you tell us like what would be like an example on that one where there would, or is that not possible?
So yeah, so the way that these 3 characters are mapped, every single one of them is mapped as a, oh, maybe this one was missed. Yeah, so we're asking which of the following characters would be a synapomorphy, represents a simplesiomorphy for a clade composed of A, B, and C, right? So let's take a look at that. So character 1, right, is a synapomorphy for all of them, or for all of the species. Character 2 is a synapomorphy for B, C, D, E, F, and G. Character 3 is a synapomorphy for C, D, E, F, and G. Right. So I think also this one actually looks like, as I'm reading it, I think that I had changed it from a previous question and I didn't change it enough. So that maybe is why you're misled. So the correct answer, I think this should have been synapomorphy here. So that's another one I've got to fix for the grad course. That one snuck
Yeah,
through.
I would appreciate it. But
does that make more sense?
The correct answer, yeah, but the correct answer would have been A. Like, would it
Well,
be character 1? Because I'm just trying to learn for the quiz.
No.
So for the exam, sorry.
No, that's fine. So let's change it back to the way it was. This is a poor question for this reason. And that's why I'll go back and give credit for on quiz 2. And I'll put a notification also. Let me make a note here so I don't forget after we get done. So this is a poor question because there are no simplesiomorphies. So if we were to have a simplesiomorphy for A, B, and C, it would need to be something that was gained in an ancestor, but then lost in one of the descendants. So for any simplesiomorphy, you would have to map the character on there in 2 places. So for instance, if it was gained, if we had a character that was gained in the ancestor of A, B, and C, but then C lost that trait, then that would be a simplesiomorphy. But we'd have to have a character mapped on there twice to qualify as a simplesiomorphy. So that's why it's a bad question. I'll change it and then I'll give credit to people. Because there was no correct answer, I'll just give full credit to everyone for question number 1 on this quiz.
So I'm sorry, I just want to get this right. So where would the node be for the correct answer for that? Or where would the line have to be? Would it be like, would it have to be like 2 lines? I said it has to be 2 characteristics.
Yeah, so let me let's see if this will allow me to kind of illustrate it. I can draw on this a little easier. Okay. All right, so let's just do a quick phylogeny here. We'll do a fairly simple one, and we'll say species A, B, and C. Now, sorry, and D. We got 4 species. Now, let's say we had a character that C and D shared, right? We can map that right there as a synapomorphy, okay? And if we had another character that B, C, and D shared, but A, and then just so it's not an apomorphy, so there's an alternative one, let's put one more and we'll call that O for outgroup. So let's say B and C share a characteristic, okay? And none of the others have it. So there are two ways that you could map that. You could map it as convergence, right? Does that make sense? Let's get rid of that one too.
Yeah, that's good. Yeah.
Yeah, so I'm going to just get rid of that first one I marked on there just so we're not confusing characters. So convergence, right?
Mm-hmm.
But I could potentially also,
Yeah.
and from a parsimony framework, it wouldn't matter. I could mark it as it happened here, and we'll say this is character number 1, and then it was lost in D. And so that's why B and C have that same character state, because some trait, let's say this is an A turning into a T, so they both inherit that T from the ancestor, but then there's a second mutation that changes it back, so the T changes back into an A, and so species D has an adenine at that site. So that would be a simplesiomorphy for species B, C, and D, because it happened in the ancestor, but then was lost in one or more of the descendants.
Yeah.
Does that make sense?
Okay.
Yeah, so that's why that was a poor question. I messed up on
That's
that
great.
one. I'm glad you caught it.
Oh no, it's okay.
Sometimes I find that I have, what's it called? When I'm writing quizzes, I have to be a little careful because I have, I forget the condition called when you're expecting this, cognitive bias is what it's called, which is mean you tend to see things that you expect to see. And so sometimes I'm proofreading my quizzes and I'm going through them quickly, and I thought that I put synapomorphy there, but it was simplesiomorphy, but in my brain I probably still read it as synapomorphy because that's what I was expecting to see. So I will change that and update it.
All right, that sounds great. And then for the term outgroup, so I'm trying to understand. So outgroups are basically, is that like your control, or is that like your known, or
A
how
little
would you describe
bit.
like an outgroup?
You might want to. It's not really like it's not proper to call it a control in the same sense that you have in like lab or experimental conditions. But in some ways, it serves as a comparison that you can compare all your other species to. So, for instance, if I wanted to understand the relationships among the birds, I would need to have something to determine, okay, what birds are, which nodes are at the bottom of the tree and which nodes are at the top of the tree. And so to do that, I pick an outgroup, which is something that is closely related to the species that I'm studying, but not part of the same group. And that's why we call it an outgroup. So based on your understanding, we'd use this as an example in all my classes. So it's fairly well known, but if you're doing a phylogeny of birds, what would be a good outgroup to choose?
It would be like a very well known bird, like a model.
No, because that would be inside somewhere. So even a very well-known bird could potentially be somewhere in the middle, and we want something that we're almost certain, and there's a little bit of a paradox or a circularity argument here, but we want something that is not part of the group we're trying to figure out, but we want a close relative, right? So technically, anything that's not a bird could be an outgroup. I could pick a tree, but a tree is so distantly related to birds that it's not really going to be useful in helping us figure out where the root of that group, the bird group, is. So we ideally would like something that's not a bird, but is as close a relative as possible to the birds.
All right, yeah, that kind of makes sense.
Yeah, and so
just so it'd be like a parameter to know where you're not supposed to go.
yeah, and so basically, so the idea is that probably for birds, your very best choice would be a crocodilian, because all the other closer relatives are extinct, right? We have all sorts of dinosaurs that are more closely related to birds, but they're gone, and so if you're using genetic data, you need something that's alive. And so the crocodilians are the closest living relative to all the birds, and so crocodile would be our best choice for, or alligator or caiman, one of those groups, would be the best choice as an outgroup for our bird analysis. And if we don't really have a good indication, if maybe this is where the catch-22 or circularity. So let's say we're doing a phylogeny for bats. And this was historically, it's a little better now, but historically was a problem because bats were so different from other mammals that we didn't know what their closest mammal relative was. So if I want to do a phylogeny of bats, usually what people would do is say, well, we've got maybe 5 or 6 candidates that could be the closest relative, but we don't know. So we'll include all of those, and those as a group will be our out group for the bats. Okay, what else, guys?
I just
All
had a,
right. Oh,
sorry.
go ahead, Nia.
I just had a quick question about the exams. They're just going to be through Blackboard, right?
Yes, so there is a time limit, but other than that, they're pretty much the same as quizzes. On the quizzes, I do put a time limit, but if people go over, as long as it's not, every single time they're going over, I still give them credit for the questions they answer late. But there will be a time frame, so it's more like a regular exam in that way. But you do have, your notes, whatever resources you want because it's online. But you're not going to be able to spend 4 hours studying and doing it. So use the same strategy as you would for an in-class exam, but with the added help of having notes or lectures or other things there. But you're not going to be able to spend 10 minutes on every question searching and finding the answer. You do need to know it fairly well ahead of time.
I actually had your like 3301, I think,
3301.
biological evolution class last semester. So
Okay.
I was kind of familiar with the exams from.
Yeah. And it will be somewhat similar to that format. The only difference is are you in the 4330 or the grad course now?
Yes, the 4330.
Okay, yeah. Yeah, so very similar to that.
Okay, thank you. Bye, Dr.
Yep.
Terry.
Thanks for coming.
So I had a question about project 1 and about project 2. I was kind of concerned if during our exam are we expected to blast anything?
No, I won't make you do that. But I might ask, it's possible that I could ask a question that relates to blast. So for instance, you might see something like, if you had a sequence of unknown origin, what would be the best strategy for determining what species or what gene it's from and what species it comes from?
Mm-hmm. And then for project 2, when is that going to be? Has that already passed? I know the
No,
old stuff.
I changed the date for it. It's due a week from today, actually. And I've nearly got it done. I'll probably post it today, but it should be quick. You should be able to complete it in an hour or less. So you don't really need to worry about it till after you get your exam done.
And if we could get, do you think we'll have the grades for project 1 before the exam so we can use it to like help us with our notes for our studying, or do you think they won't?
Yeah,
Project 1 won't really
I
have
should
much?
have that this evening. I'm going to start grading them, and it's kind of big for a grad course, but I should have them done by early tomorrow morning.
All right, that sounds great. Thank you.
Yep.
Hi, Dr. Terry. So yeah, I just want to thank you for answering our questions. So it helps us a lot. So yeah, thank you.
Good. No worries. Yeah, that's why I'm here.
Okay. Bye, Dr. Terry.
Yep. See ya.
See ya. Bye.
Yep. And then Vanessa, I'll post this. I'll have to figure out how to do it. I've not recorded a Zoom session before, but once we get all done, I'll post this and I'll put a link and also notification regarding that quiz 2 question.
And also for Nia's, were you going to change that too? I don't remember which one that was in. I think it was in quiz 4.
Yeah, but that was for, I think that was for the 4330 class.
Oh, that's the undergrad?
Yeah, that was the undergrad. The material is quite similar at this
okay.
stage. Yeah, they go, so they're kind of parallel courses. And I think even some of the grad students may have had that at the undergrad. If so, there's a lot of review between the two courses. Yeah, but
okay, because I heard Nia say that she did take your undergrad course and that now she's in your grad course.
Yeah, but I think that Nia had taken the 3301 course, which is the introductory.
Okay, yeah, I just wanted to make sure.
Yeah. Yeah, but I have had some
Well,
students. Sorry, I have had some students who've taken my upper division undergrad course, and then they go on to take the grad course also, and there's a lot of overlap. So I don't know if that's boring for them or if it's nice because they know a lot of the material already.
Yeah, and they get to relearn a little bit more of it.
Yeah, and reinforce, and yeah, so maybe that's not so bad, especially when you're dealing with lots of stress and other things in other classes.
Do you think it's helpful? Because I have your evolution textbook still, and I also I know the author, he's done really good work. I think he wrote a book called I think you mentioned it, it's called You Can Hear Her Mother in Her Laugh or something, and it has to do
Yeah.
with I think it has something to do with CRISPR.
Yes.
Do you think that literature is helpful for this class?
Yeah, we'll talk briefly about CRISPR near the end of the semester. That's a little bit more of kind of biotech and hands-on, how we change things. So it's not really related to what we do in this class, but yeah, I mean, the general underlying kind of factors of what's going in when people do that always will have a phylogenetic basis, or at least they should if people want to make it easier. So if you want to make changes, for instance, to figure out what's going on, you need to understand the diversity that's already there and how that has impacted evolution, and make sure you're not, either doing unnecessary work or making a mistake or causing problems. So certainly what we're doing here is a foundation and a background for those. And if you understand those principles, yeah, that'll help. I wouldn't look at it and say, oh, I need to spend a lot of time on this so I can do well on the test, but certainly reading it and applying the principles in those ways will exercise your brain and help you do better, when it comes time to making those leaps of understanding.
Yes, that sounds good. Alrighty, it's already 4 o'clock. I have to go.
All right.
Well, you
Thanks,
have a good
Vanessa.
one, Dr. Terry.
Yep.
All right,
See ya.
bye.